Hello, welcome back to the channel Awesome Learning. The next topic from the chapter Biotechnology and Applications is transgenic animals. So let's begin with this topic. These are the animals whose genome has been altered by introduction of an extra or foreign gene by manipulation. So I hope you have now understood what do you mean by transgenic. When we try to change or when we try to alter the genes, so these are called transgenic animals. Example, transgenic rats, rabbits, pigs, sheep, cows, fish. Okay, over 95% of transgenic animals are mice. So we have seen so many examples here. Now let's see benefits. Why do we want to do this? Why do we want to transgene the animals, or we want to alter their genetic codes? So number one is to study the regulation of genes on normal physiology and development. Now we want to study various aspects of how the genes regulate the normal physiology. Now you have studied the full topic of regulation of genes. We know that genes have the role of regulation. They control which enzyme or protein will be formed, how much it will be formed, what will be the purpose or what will be the function of those enzymes and proteins that will be done in the body. So to study all these things we need to do this transgenic a technique of transgenic in the animals. Second is to study the contribution of genes in the development of a disease. Of course, we want to find out a particular cause of disease. Then we also want to find out the treatment or prevention of those diseases. So how are genes used, or how can they be used to in the purpose? This also has to be done. And so the technology of altering the genes in animals is being done. Then biological products, we actually are dependent on animals for many products which are used by humans, either you can see as a medicine or as a particular product to consume. So we are depending on them. So for those also we need to alter the genes of the animals. Next is vaccine safety testing. Of course, if we have developed a certain vaccine, we just cannot go and inject those vaccines into human population. So we need to first test those vaccines on the animals, and then it undergoes lot of steps. And if all the steps are fine, and if the boards have agreed to it and the countries have agreed to it, then only it will come into the market. We are still waiting for the vaccine for COVID-19. Next, chemical safety testing, that is toxicity testing. Suppose a particular chemical has been used in the market, but suddenly we see that the chemical has been hampering the health of the population, either of animals or humans. So just to test those chemicals, we can do this in animals. So the altering of gene is to be done in the animals. So these are the various reasons why what is the benefit of transgenic animals. Now here in this topic, first the regulation of genes on normal physiology and development. The first topic that we will see is examples. Now children, here these examples are very important because they will come to you as a question. So study of complex factors such as insulin-like growth factor, genes from other species that alter the formation of this factor are introduced, and the biological effects are studied. So such factors are introduced from any other species. Suppose there are species A and species B, and we have taken the factors from species A and we have introduced it into the other species, and we just want to study that what will be the result of this new factor being added into the new species. This gives information about the biological role of the factor in the body. So insulin growth like factor 1, right? We have done this in humans when we were not being able to produce insulin. What we were doing, We were just taking the insulin from the cattle or the pigs, and then we were introducing it into the human population. And we also saw that due to this, there were certain reactions which were seen in the human population. So all these can be done here. Then the second one here is the see transgenic models of help to investigate new treatments for human diseases. Example, transgenic model for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. These has been studied, that diseases have been studied, and we had used transgenic animals. We want to find out the treatment of these diseases, so we are using transgenic animals for making the experiments or for doing the experiments, so that we can come out with the proper drug or the way of prevention. Third is biological products. These animals are used to produce biological products by introducing genes which code for a particular product. For example, suppose we want the human protein alpha 1 antitrypsin. Now this is a product used in the treatment of emphysema. So this is used in the treatment of the problem called emphysema. So the product of treatment of phenylketonuria and cystic fibrosis also has been taken out from the transgenic animals. So these are the products which we get from transgenic animals. In 1997, Rosie, first transgenic cow, produced human protein enriched milk. So you just see that the milk is rich in human protein, 2.4 gram per liter. 2.4 gram of protein was found in 1 liter of milk. So it contains human alpha lactalbumin. This is the human protein found in the cows, and it is nutritionally more balanced product for human babies than the natural cow milk. Okay. So this is again a product, biological product. You will have to just learn by heart, because these can come in your 1 markers. Then vaccine safety testing. Transgenic mice are used to test the safety of the polio vaccine. So we used these mice to test how the vaccine is, whether it is safe or not, before we wanted to use it for the human population. So if it is reliable, they can replace the use of monkeys. After rats, this will be done on monkeys, and then after safety, if it is safe in the monkeys and apes, it will be then taken to the human population or human babies. Then chemical safety testing, that is toxicity testing. Some transgenic animals carry genes which make them more sensitive to toxic. Okay, so substances that than non-transgenic. Suppose there is a substance A, and it has been introduced into both transgenic and non-transgenic. So transgenic will be more sensitive to this, and non-transgenic will not be sensitive. And if the transgenic animals die or they show certain reactions, हमें पता चल जाएगा कि जरूर इस प्रोडक्ट में कोई चीज है जो टॉक्सिक है, क्योंकि हमारा transgenic animal जो है वो बहुत sensitive है. They are exposed to toxic substances and the effect studies, it gives immediate results. So this is all about transgenic animals.